Welcome to G Fun Facts Online, the show that unpacks the science behind the headlines. I'm your host, and today we're diving into one of the most exciting and challenging fields in all of medicine, gene therapy. Imagine being able to fix diseases at their very source, rewriting faulty genetic code like a programmer fixing a bug in an app. That's not science fiction. It's the promise of gene therapy. It holds the potential to cure everything, from rare genetic disorders to common killers like cancer and heart disease. But there's a catch, a huge one. How do you get the cure, this piece of genetic code, to the right place in the body? You can have the most brilliant cure in the world, but if you can't deliver it, it's useless. This is the delivery challenge, and it's the central puzzle that thousands of scientists are working to solve right now. Today, we're going to explore the two main contenders in this high-stakes delivery game: nature's own experts, viruses, and the custom-built challengers, nanoparticles. It's a battle of biology versus engineering, and the winner could change medicine forever. So what exactly are we trying to deliver? At its heart, gene therapy is about introducing new genetic material into our cells. This could mean replacing a broken gene, turning off a gene that's causing trouble, or adding a new gene that helps our bodies fight disease. But our bodies are designed to be fortresses. A therapeutic gene has to survive a journey through the bloodstream, avoid being destroyed by our hypervigilant immune system, find the exact right city of cells in the vast country of our body, knock on the right door, and get all the way to the city hall, the nucleus, to deliver its message. It's an incredible biological obstacle course. To navigate it, we need a vehicle, a vector. Think of it as a microscopic Amazon Prime driver for our DNA. For a long time, the most obvious delivery drivers were viruses. After all, this is what they do. Viruses have spent millions of years evolving to become unbelievably efficient at breaking into cells and inserting their own genetic material. So scientists had a brilliant idea. Let's hijack them. They learned how to remove the harmful, disease-causing parts of a virus and replace them with a therapeutic gene. What you're left with is a recombinant viral vector, a disarmed virus that's now working for us, delivering a cure instead of a cold. Let's start with the current celebrity of the viral vector world: adeno-associated viruses, or AAVs. If viral vectors were cars, AAVs would be the reliable, super safe family minivan. They are incredibly popular because they're not known to cause any disease in humans, and scientists have made them even safer for therapeutic use. One of their biggest advantages is safety. They generally don't shove their genetic code into our own DNA. Instead, the gene they deliver floats around inside the cell's nucleus as a stable, separate little circle of DNA. This drastically reduces the risk of accidentally breaking one of our important genes, a problem we'll talk about later. They also trigger a relatively mild immune response and can provide long-lasting expression of the new gene, sometimes for years. But the AAV minivan has its downsides. The biggest one is its tiny trunk space. AVs are small, and they can only carry a small genetic payload, typically less than 4.7 kilobases. This is fine for some diseases, but if the gene you need to replace is a big one, it simply won't fit. Another huge challenge is pre-existing immunity. AAVs are common in the wild, so many of us have already been exposed to them and have antibodies ready to attack. If a patient has these antibodies, the gene therapy can be neutralized before it even gets to work. It's like the delivery address is already on a do not deliver list. And even though they are low-key, they can still trigger an immune response, which might prevent a patient from ever receiving a second dose. Finally, making enough high-quality AAVs for everyone who needs them is a complex and very expensive manufacturing puzzle. Now, if AAVs are the safe family minivan, then lentiviruses are the heavy-duty pickup trucks with the power to modify the whole garage. Lentiviruses, which are a group that famously includes HIV, have a special trick up their sleeve. They permanently integrate their genetic material right into the host cell's own DNA. Scientists, of course, have created safe, replication-incompetent versions derived from HIV that cannot cause AIDS. The massive advantage here is stability. Once that gene is integrated, it's there for good. Every time the cell divides, the therapeutic gene is copied right along with it. This is crucial for treating diseases that affect dividing cells, like certain blood disorders. They also have a bigger cargo capacity than AAVs, able to carry larger genes. But this superpower of integration comes with a serious risk: insertional mutagenesis. Imagine you're trying to add a new sentence to a book, but you write it randomly over an existing crucial paragraph. Integrating into the genome is random, and there's a small but real risk that the new gene could land in a bad spot, disrupting a vital gene or, even worse, activating a gene that causes cancer. While newer generations of lentiviral vectors are designed to be much safer and avoid these hotspots, the risk isn't zero. The public perception can also be a hurdle. The link to HIV, however scientifically unfounded in the therapeutic context, can cause concern. Then we have the veterans of gene therapy, adenoviruses. These were some of the first vectors ever used. They are incredibly good at their job, able to infect a wide variety of cells with high efficiency, and they can carry large genes. But they have a major public relations problem with our immune system. They are known to provoke a very strong immune response. It's like they show up with sirens blaring and lights flashing. This was a key factor in the tragic death of a patient in a 1999 gene therapy trial, which set the entire field back for years. This strong immune reaction means the gene expression they provide is usually short-term, as the immune system quickly clears out the treated cells. This makes them less suitable for chronic diseases that need a long-term fix. Because of all these challenges with viruses, the safety concerns, the immune responses, the manufacturing headaches, scientists began to wonder, can we build a better delivery vehicle from scratch? This brings us to the engineered alternative, nanoparticles. And the undisputed star of this world is the lipid nanoparticle, or LNP. If you've had a COVID-19 mRNA vaccine, you've already experienced LNP technology. They are the unsung heroes of the pandemic. So what are they? Essentially, they are microscopic bubbles of fat. That's it. These lipids or fats are engineered to wrap around a delicate genetic payload, like mRNA, protecting it on its journey into the cell. Think of it as a tiny, greasy envelope for a genetic letter. The advantages are huge. Because they are synthetic and not derived from a virus, LNPs are generally much less likely to provoke our immune system. This means they could be used for therapies that require multiple doses. They are also much easier to manufacture at a massive scale, as we saw with the vaccine rollout. And they're versatile. You can package all sorts of things inside, from mRNA to the gene editing machinery of CRISPR. But LNPs aren't perfect. Their biggest challenge is targeting. They have a natural tendency to accumulate in the liver. This is fantastic if you're treating a liver disease, but it's a major limitation if you need to get your therapy to the brain, or the heart, or the muscles. Scientists are working hard to stick molecular address labels on them to guide them to other organs. But right now, viral vectors are still the champs of precision targeting. Another hurdle is efficiency and what's called endosomal escape. After an LNP is taken into a cell, it gets trapped in a little bubble called an endosome. The genetic payload needs to escape this bubble before the cell destroys it. Making LNPs that are good at this jailbreak is a key area of research. So let's put them head to head. Viral vectors generally offer higher delivery efficiency and more specific targeting, with the potential for very long-term gene expression. But they come with a limited packaging capacity and significant safety concerns about the immune system and, in some cases, the risk of cancer. Nanoparticles, on the other hand, are generally safer, less immunogenic, easier to make, and more versatile in what they can carry. But their targeting is less precise, and their efficiency can sometimes be lower. It's a classic trade-off: the evolved, high-performance but risky natural system versus the safer, more flexible but still in development engineered system. The future of gene therapy depends entirely on overcoming these delivery hurdles. The first major frontier is taming the immune system. We need to make these vectors invisible. Researchers are trying to engineer viral capsids and nanoparticle surfaces to be stealthier, or using drugs to temporarily and safely suppress the immune system during treatment. The second frontier is targeting. We need to turn these delivery systems into microscopic guided missiles. This involves engineering them with molecular keys that only fit specific locks on the surface of target cells, or designing smart nanoparticles that only release their cargo when they sense the unique environment of a tumor. And finally, there's the challenge of scale. As more of these therapies are approved, we need to be able to manufacture them consistently, safely, and affordably for millions of patients. This is an enormous industrial challenge, moving from a bespoke laboratory process to a global manufacturing pipeline. So what's the final verdict? Virus or nanoparticle? The truth is, there probably won't be a single winner. The future of gene therapy will likely be a diverse toolkit, where doctors choose the best delivery system for a specific disease and a specific patient. Maybe an AAV is perfect for a non-dividing tissue like the eye, while an LNP is better for a therapy that requires repeat dosing, and a lentivirus is needed for a permanent fix in bone marrow. The most exciting part is how different fields are converging to solve this problem. Nanotechnology, virology, and even artificial intelligence are coming together. AI and machine learning are now being used to design brand new viral capsids and nanoparticle formulations from the ground up, properties that would take years to discover through trial and error. The delivery challenge is immense, but the progress is breathtaking. As we get better and better at building these microscopic couriers, we move closer to unlocking the full, revolutionary potential of gene therapy, and closer to a new era of medicine where we don't just treat diseases, we cure them at their most fundamental level. Thanks for tuning in to G Fun Facts Online. Join us next time as we explore another fascinating corner of the scientific world.